So in this video I am going to discuss you about the microscopic features of ulcerative colitis and Crohn's disease. But before discussing these microscopic features, I want to discuss you about the histophysiological diagrams of intestine, because these ulcerative colitis and Crohn's disease these are the pathological conditions, so it is better before we know the normal findings. So therefore, let's begin with the histophysiological diagrams of intestine. See the normal histophysiology of intestine. So first of all, as you can notice that there are 4 layers. So this is the mucosa, and this is the submucosa, and this is the muscularis mucosa, and this is the serosa layer. So there are 4 layers of intestine now, and this is the lumen. So you can notice that this mucosa of intestine, so here extends into the lumen, and that elevation of that mucosa into the lumen of the intestine so leads to formation of villi, and also that is a depression of that mucosa again to reach the normal line layer is known as so here crypt. And you can also notice that in the crypt also that there is a gland known as crypt gland is present, and also you notice that in the lumen of the crypt gland there are no cells are found. And also see that, and this is the Peyer's patch. So this is the Peyer's patch you can notice, which is also found, and this Peyer's patch is just so here accumulation of normal lymphocytes. So therefore, so what are the normal histophysiological findings? So that the 4 layers, mucosa, submucosa, muscularis mucosa, and serosa, and also villi, and this villi is the elevation of mucosa into the lumen, and the crypt is just so here depression of a mucosa crypt, and also so here crypt gland is found, and in the crypt gland there is a lumen, and in this lumen no cells are found, and this is the Peyer's patch, and this is just so here accumulation of lymphocytes. See, all points are written. So see that 4 layers present: mucosa, submucosa, muscularis mucosa, and serosa. Next, elevation of mucosa layer into the lumen is known as villi. The third, depression of mucosa layer into the normal or to reach the normal line is known as crypt. And in crypt gland is also found, and in the lumen of the crypt gland that is no cells are found. And fifth is the Peyer's patch found. And what is Peyer's patch? And this Peyer's patch is the accumulation of lymphocytes. These are all the normal histophysiological findings of intestine. And in the histopathological diagrams, you will also find these structures and along with some abnormalities. So this is the histopathological diagram of ulcerative colitis. So once again, that is, what are the 4 layers of the intestine? So this is the mucous layer, submucous layer, muscularis mucosa, and this is the serosa layer. Now, normally, as you know that in the intestine, that in the histophysiological diagram, the mucosa layer is in continuous. So there is no discontinuation between the mucous layer. But in this case, you can see that in this region that there is a discontinuation between these 2 mucous layer, and this discontinuation of mucous layer is done by ulcer. So in the histopathological diagram, so ulcer is found in the region where there is a discontinuation between the mucous layer. So here then this ulcer is found. Now you can see that this ulcer does not extend up to the mucosa layer. That is ulcer, so it doesn't extend up to the mucosa layer. That it so here stays only up to the so here that is mucosa layer. So therefore it is known as so here that is superficial ulceration. First thing, and next you can also notice that in this so here ulcer region, so here that is neutrophils are accumulated. So here then, so these are the neutrophils, as you know that. So here normally neutrophils have 2 to 5 lobes, and in this case 3 lobes also. So here that these are neutrophils. So neutrophils are present in the ulcer region. So these are act as here inflammatory infiltrates. Now, and you can also see that so here congested blood vessels are also present in the ulcer region. So that is all about the, so here that is things on in the ulcer region. So first thing that it is superficial ulceration, and also in this ulcer region, so here that is neutrophils are present and they act as inflammatory infiltrate cells, and also congested blood vessels are found. Now, as you already know that in the crypt region, that crypt gland is found, but you also know that in the lumen of the crypt gland, that is no cells, no neutrophils are found. But in this pathological condition, you can see that in the lumen of the crypt gland, so here neutrophils are found, and that so here accumulation of neutrophils in the lumen of the crypt gland is known as crypt abscess. So in the pathological structures, what we have found, the first one is that superficial ulceration, as it extends only up to the so here submucosal layer, it does not extend intramuscularis mucosa. And second, that is neutrophils are found in the ulcer region, which act as inflammatory infiltrate cells, and also congested blood vessels are found. And next, so here crypt abscess, and this crypt abscess, that means the accumulation, focal accumulation neutrophils in the lumen of the crypt gland. And see, all these portions are written. So the microscopic features, so first one, that superficial ulceration is present. Why? That this ulceration does not penetrate the muscular layer of the intestine, or simply muscularis mucosa, you can also say. And second, that in crypts, so here focal accumulation of neutrophils present in the lumen of the crypt gland, and that accumulation of neutrophils called crypt abscess. And third, see that in the superficial ulcer region, inflammatory infiltrates are present, and these inflammatory infiltrates are neutrophils. And the fourth thing is about the so here congested so here vessels are also present in the superficial ulcer region. That's all about the microscopic or histological histopathology of ulcerative colitis. So let's discuss about the histopathological feature of Crohn's disease. So once again, so this is the mucus layer, and this is the submucous layer, and this is the muscularis mucosa or muscular layer, and this is the serosa layer. So now, as you already know that in the normal histophysiological structure, that the mucosa always remains in a continuous layer. But see that in this case, that here ulcer is present. Why? Because discontinuation occurs between the mucus layer. So here also ulcer is present, and in this region also ulcer is present because discontinuation occurs between the mucus layer. Now see that in this case the ulcer so here extends up to the so here serosa layer or the muscularis mucosa layer, and also that this ulcer gets converted into deep fissure, and since this ulcer so here extends up to the layer of so here extends up to the layer of serosa, that is it is also known as deep fissure, or sometimes it is also known as transmural. Now again see that so here also non-caseating granuloma is also found. So this is a non-caseating granuloma, so the components are also there. So this is that is a modified epithelioid cells, giant cells, lymphocytes, and these are the plasma cells. They together form the so here non-caseating granuloma. And see that, and in all the four layers so here lymphocytes are present. So this is the lymphocyte, so in the submucous layer, and in the muscularis mucosa also so here lymphocyte is present, and in the serosa layer also a lymphocyte is present. So lymphocyte is present in all the layers. So what are the So here that is microscopic features here found. The first one is the deep fissure, and this is the ulcer that is which is converted into deep fissure when it extends up to the serosal layer. The first thing is about a deep fissure, and the next is about a non-causating granuloma. So see that this is a non-causating granuloma, and the components are giant cells and the epithelioid cells and the lymphocytes and the plasma cells. So this is a non-causating granuloma. And the third thing is that see that in all the layers, so here lymphocytes are present, so that this lymphocyte is present in submucous layer, and this lymphocyte is also present in the muscularis mucosa or muscular layer, and this lymphocyte is also present in the serosa. So these are all the here that is histopathological findings. So see what I have written. So the microscopic features. So the first thing is that ulcer extends up to the muscularis mucosa or the serosa layer, and this ulcer takes the form of deep fissure, the first thing. And second, non-causating granuloma is present, and this non-causating granuloma so here consists of giant cells, epithelioid cells, lymphocytes and plasma cells. And the third thing that lymphocytes are present in almost all the layers. So these are the here histopathological findings of Crohn's disease. In Crohn's disease, you noticed about non-causating granuloma, and also discussed about the components of non-causating granuloma, what are giant cells, epithelioid cells, lymphocytes and plasma cells. But now I want to discuss how these giant cells and epithelioid cells are formed. So friends, as you already know that in the blood vessel monocytes are found, and these monocytes they come out of the blood vessels and they reach in the tissue region, and in the tissue region that these same monocytes they are converted to macrophages, and then these macrophages, that is an abnormal condition or a pathological condition, that is macrophages converted to epithelioid cells. So epithelioid cells are formed when these macrophages converted into epithelioid cells, or these macrophages undergo conversion. Second, see that so these are the epithelioid cells, and all these epithelioid cells that they do fusion with each other, and due to fusion of these epithelioid cells with one another, so here giant cell formation is done. So how this epithelioid cell is formed? So first that these monocytes come out of the blood vessel, and these monocytes reach a tissue region to become macrophages, and these macrophages again converted to epithelioid cells, and then all these epithelioid cells do fuse with one another, and due to fusion of these epithelioid cells, so here giant cells are formed. And see what I have written. See that these monocytes come out of the blood vessel to become macrophages, and second, next these macrophages converted to epithelioid cells, and third, all these epithelioid cells do fusion with one another to form giant cells. The video finally comes to an end. So in this video I mentioned about the normal histophysiology of intestine, and also what are the components found in the normal histophysiology of intestine. And second, I discussed about the histopathology of ulcerative colitis, and third, histopathology of Crohn's disease, and fourth, how are epithelioid and giant cells found in the non-causating granuloma found in the Crohn's disease.